Trojans Talk Research is a monthly spotlight on the research interests of Troy's faculty presented by Troy University's Office of Sponsored Programs. This month, Dr. Alexander Kofman will present his research on microRNAs and genetic polymorphisms in the age of human variation biology.
All right. Hi, I'm Dr. Harden. I'm the chair of the biology department. I'm excited to introduce my friend and coworker, Dr. Alex Kofman. Dr. Kofman has a kind of unique history. He started with getting his medical degree from St. Petersburg in Russia. It's one of the finest med schools in Russia. And then he wasn't done with his research at that point. He moved on to Regensburg, Germany, where he studied virology and genetics, and he earned a PhD, also from Russia, in virology and genetics. He then began his career, well, I guess he had already started really his career in the U.S. As a postdoc, floating around to many of our really fine institutions, from Columbia to Beth Israel Medical Center, and then I guess finishing up at University of Virginia. We were very excited in 2018 when he joined our staff and began his research projects. He started the Human Genetics Club with us. I think many of the people here are part of the huge club. And he also has his own interesting research that he's going to present today on microRNAs. So welcome to Dr. Kofman. All right.
Thank you very much. I want to thank first of all the Office of Sponsored Program and Leanne and Paula for this wonderful and unexpected opportunity to present my research to you. So I feel a little bit, let's say it's the first time I'm presenting here, and the equipment, you know, and the space. I will need a little bit to adjust myself, and excuse me if something will be knocked down, you know, and hopefully not the whole step. Anyway, so let me start. So first of all, this is the first slide. It's a little bit funny, and I like to start my presentation with the slide. Actually, it's also about human variation in the research world. Well, unfortunately, we have here undergrad students, we have here PhD students, right, and how they view each other. We don't have here the Master of Science students, unfortunately, but Master of Science students are also important because before it's PhD student, it will be Master of Science students, so consider. All right, and today's topic is about microRNA and genetic polymorphisms in the age of human variation biology. Let me tell you, first of all, about human variation biology. Just a few words, and in general, what kind of research it is, and how the things changed during the last, maybe, well, beginning from 2005, during the last 20 years, less than 20 years. It's a new kind of research, and it's ongoing revolution, what's happening now. No, human variation and predisposition to various human diseases. This is the most important, since it's Department of Biology, and I'm a medical doctor, and we are doing biomedical research. We are trying to do biomedical research, right? So many things, as you can see. Well, the sex, the age, the marital status, even the marital status, the educational status, the work and working environment, the income, nutrition, housing, and environmental, generally environment, you know, where we live in, right? And even comorbidities and genetic traits, it's all matters. It's all matters, right? What I mentioned here are actually the social factors, but they also matters. They also contribute to predisposition and the probability of developing certain disease and the health status, right? No, well, human variation biology and social aspects. We are talking about variation. You remember what was happening just a few years ago, and the same thing was happening 100 years ago during the influenza pandemic of 1918 and COVID-19 pandemics. So the sign here, well, I took it from online. I knew this image, right? It says, wear a mask or go to a jail. Why such a level of argument? Because we are all different. Some of us can, well, we all were infected by COVID-19, but only few of us would become sick, and few of those, the fraction of those, would die, while others not. It's because we are different. We are different, and it was a big question. I don't want to go to politics and ethics of this, but you see how much it's important. The variations, the human variations, biological variations, may even shake up the society. And the same things were happening 100 years ago, so nothing new today. Now, if you talk about human variation biology, it's not something new. About 60 years ago, it was noticed that the genes that belong to the so-called major histocompatibility complex, and this is about immunology, and in humans, these genes are called human leukocytes antigens, HLA. These genes, maybe many of you heard about them, right? Even if you didn't study immunology. It's about immunocompatibility of tissues. It's about something to pick up the right donor and the recipient of organ and tissue, right? So that's what we know these genes for. But at the same time, as early as 60 years ago, and I remember I was reading this publication, it was noticed that changes in these genes, and these genes are actually highly polymorphic genes. That means they are characterized by they are highly variable. You know, the primary structure of these genes, the sequence of nucleotides, the primary building blocks may be different. And variations in these genes, here they depict like they're all located on the 6 chromosomes, right? And there are 3 major segments, and this more in details. It's not about my topic, but I just wanted to bring it, right? The variations in these genes may be linked with predisposition to various diseases, starting from autoimmune and inflammatory disease, and including even type 1 diabetes, which is, you know, which is kind of, the recent studies show that the type 1 diabetes may be caused by the infection of the Coxsackie B virus. So, you know, it's about infection, but still it's autoimmune disease, right? And the structure of these polymorphic genes, HLA genes, major MHC genes, it may somehow influence the development of this disease. And gastrointestinal disease and even neurological disorders, the Parkinson's disease, which is very actual for studying, right? And even psychiatric disorders, including schizophrenia and autism. Let me stress that these are the so-called multifactorial diseases, the diseases that are known, according to medicine, then they may be caused by different factors, or rather combination of various factors, but including genetic predisposition. And this is the HLA genes. Yes, and they are known, they are linked. Certain types of HLA genes, of this major histocompatibility complex genes, certain types are linked, associated with a higher probability of developing even schizophrenia and autism, and joint disease, and human cancers, and adverse drug reactions, and response to vaccines, hepatitis B, and even male infertility. And once again, and it's about infections, including the development of HIV infection and HBV, human hepatitis B infection. So it was well known and is being studied, and mechanisms are not clarified. But in today's topic, I will show how the things changed and what's new now in this field of human variation and predisposition to certain disorders. Now, let me tell you that what we live at the time of ongoing revolution, and the most important event of the century, if you want, was completing the Human Genome Project. So we know all genes in all the chromosomes. We know the primary sequence of the genes, that is the sequence of nucleotides, the primary building blocks. And this project was completed with the classical Sanger sequencing method. It allows to sequence at once it could be one fragment of DNA, length up to 900 maximum bases. And using this technique, the whole human genome was identified, deciphered, right? Now, since we know which gene is located on each chromosome and in which position, we are talking about the normal genotype, normal human genome, right? And we are not talking about translocation, chromosomal translocation that may happen, you know, as events after the ionizing radiation and genotoxic stress, right? Then The next step was around 2005. Well, let me see complete. Yes, it shows around 2005, second generation sequencer, but where is the first generation sequencer? The first generation next gene sequencing was developed around 2005, and it was the new type of sequencing, opposite to Sanger sequencing. It would allow to sequence rather short fragments of about 50 nucleotides only, but in millions of fragments. And since we know the structure of the genome, the computers would allow to reconstruct the whole genome after such sequencing. So it would allow to sequence the whole individual genomes at once, basically at once. And what's happening nowadays, this is the ongoing revolution, and everywhere, all around the world, and here at Hudson Alpha Research Institute in Huntsville, they have the room the size of this class, and in the room there is dozens of sequencing devices, and they sequence all the genomes obtained from normal people, from the patients, and some devices allow to sequence several samples at once. And this is what's happening, and this is the revolution, because we can learn a lot about our differences, not just at the level of the MHC complex, but other differences, and we have plenty of differences. This here. And as a result of the sequencing, the biologists start dealing with the so-called big data. So the big data are data, a huge amount of data that may change, because once the new sequencing results are coming from the different regions of the world, and of course we know there are ethnic and racial disparities in the genomes, and this data are being accumulated, but it's difficult to analyze them, right? It's difficult to structure the data so that can be used easily, right? We are talking about the big data. And the system biology that used to talk before is kind of, this is the field of biology, and this is the legitimate field of biology that, that is dealing with the analysis of the accumulated data. So no experiments are done, but the data are being analyzed. And it's not something new. The discovery of DNA, Watson-Crick model, they did not do any experiment. They just analyzed the publications, and they received the Nobel Prize. It was a matter of some disappointment for many scientists. So this is the classical example of system biology, analysis of the data. They did not have too many data. Yeah, it was just few publications. They managed to analyze them, and they built up the DNA model, right? And other examples, for example, the so-called Kozak sequence, I usually put them, since I used to teach molecular biology, this is the beginning of the protein encoded, and it's the same sequence. It's conserved sequence for hundreds of human genes. This is also example of the system biology. No experiments were done, just analysis. Around 1980s, she published this paper, Marilyn Kozak, she's an American scientist. The analogous situation with Shine-Dalgarno sequence when it comes to bacteria. So this is example of system biology. But nowadays, we have plenty of data, and we need somehow to find something interesting in this data. And there is a term, legitimate term, that's called data mining. Data mining means that we have a lot of data, usually in databases, and we need to find some pattern. It's not about downloading data from the database to your laptop, right? And say, well, I did research, I download. No, it's analysis of this data and finding some patterns. And this is what we are doing here. And I will show you later how, right? So this is called data mining. It's a new terms. They are resulting from the ongoing revolution of the next gene sequencing, of generating the mass of data, right? And this is the bioinformatics, of course, and we cannot do it. We can do nothing without computers and appropriate tools. And the first such tool was developed in the 1970s. It was classical tool, alignment of two nucleotide sequence. And since then, bioinformatics is being developed and developed and developed. It's called bioinformatics, biocomputing. This is also reflects the ongoing revolution. Now, what is all about? What we have today? Well, it's about computer modeling upon the existing experimental data. It's about prediction, what we may expect if you have this mutation or this variation, right? It's about mathematical approaches to biology. And finally, ultimately, it's about precision medicine, or so-called individualized medicine, when upon the analysis, upon the knowledge of the genome of the specific patient, the specific therapy will be appointed. And even, well, Ms. Janet Gaston said, and even in case of nutrition, before recommending the certain diet, first of all, the analysis is done. So this is the individualized, personalized medicine. All right. Now, human genomic variation. What is the focus of today's research? Assume that, well, this is the chromosome and the number of nucleotides. It's not about big changes, because big changes are usually associated with pathology for sure. It's about small changes at the level of the single nucleotide. Such small changes may stay unnoticed. Nobody knows. But these changes may influence. They may happen as mutations. They may affect the protein, or they may just stay unnoticed. But one of such changes, or more probably several changes, coinciding, they may have some effect. It may be a factor predisposing our humans to some disease, or maybe makes us resistant to some disease, which is more difficult to prove, because if you're not getting sick, that means nothing happened. If you're getting sick, yes, then maybe some studies, the so-called genome-wide association studies, to see how many people develop this disease and how many people have these certain specific changes, right? Yeah. So this is the single nucleotide polymorphism. Once again, changes at the level of the single nucleotide. And this is just change of one nucleotide. It may be mutation, maybe not. It may be unnoticed. This is the focus of today's human variation biology research, right? Now, this is how SNPs look like on the genomic map. The red ones are substitutions of the nucleotides, and nucleotides are buildable. The blue ones are deletions, removal of nucleotides, or insertions. Just like that, right? Now, I'm coming, after this introduction, I'm coming to our project to explain what we are doing and why we are doing this and what we are trying to do here. And lecture 2, well, lecture 2 because the students who are taking genomics class, I already told this, and you are sitting and you feel a little bit boring because we were talking through this again and again, but keep in mind, there will be tests on this, so it's just a chance to go through this again and be ready to the test, right? So we are currently working on 2 factors that may influence microRNA functions, and I will tell you what the microRNA is. It's about homologous microRNA binding sites and single nucleotide polymorphisms in the microRNA binding sites. Let me start with small explanation. So this is our gene that produces the so-called messenger RNA. Our genes produce messenger RNA and send them to ribosomes to direct the synthesis of the protein, right? So each messenger RNA consists of the beginning, which doesn't have information about the protein, the main part, the protein coding sequence, right? And the rather long 3' untranslated region, the tail, right? And you know this slide. So here, this is the promoter region. The changes here, well, the promoters are pretty much resistant to changes. And you need really to cut off the good piece of the promoter to see some change. And it will be about the amount of protein produced. In the protein coding region, the situation is not good, right? Well, the changes in the amino acid sequence, we used to call them mutations, dysfunctional protein, right? Well, there may be also other changes that influence the amount of protein produced. By the way, I should say that at the age of human variation biology, it's better, you know, there is a tendency not to say mutation, because nobody wants, you know, to be referred to as a mutant, right? Rather, we talk about variants. So let's say that the variants here, they influence the amount of protein produced. The variants here, they may influence the structure of the protein, and the protein may become dysfunctional, and it's very obvious, and there may be a lot of cases. Here, in this area, no proteins. The variants here, well, it may influence the amount of the protein produced. And there are many proteins that bind here and regulate gene expression by letting messenger RNA exit out of the nucleus to the ribosome, to the cytoplasm, or stay rather hold it in the nucleus. And this so-called microRNA, they also binds to this, and microRNA regulates gene expression. You know this, right? So this about microRNAs. They are short, non-coding RNAs, usually around 21 to 25 nucleotides. What do? They are present everywhere, in all the biological species, and even in the viruses. In bacteria, formally speaking, they are not called microRNA, but they do analogous functions in all the biological species. And they regulate gene expression by suppressing, as generally accepted. Well, I know there are examples, but generally speaking, they bind to the messenger RNA upon the complementarity. And you know what the complementarity is, right? It's Watson-Crick complementarity. And they cause degradation of the messenger RNA, or rather they hold it and prevent messenger RNA loading onto the ribosomes. So they exert negative action, but they are important. So these binding sites, which is typical for mammalian cells, in plant cells, they bind along their length, and there is complementarity, but as we know now, in mammalian cells, it doesn't work, because in mammalian cells, the full-length binding of the microRNA may cause its degradation. So there is difference between mammalian cells and plants, right? But in mammalian cells, this region, the so-called seed region, is very important. It should match perfectly. It shouldn't be interrupted. In this case, microRNA is functional. Now, we are talking about variations. We are talking about variants. And everything may happen here and here and even here, and there may be some change here, right? And then there is no full-length match, and what may happen? Let's take a look at this image. This is the classical image, right? Assume we have the oncogenic messenger RNA, and it's under the control of the microRNA. That's how it looks like, right? Full match. So this microRNA keeps, well, prevent synthesis of too much oncoprotein, right? And this, in case of polymorphism, we call them single nucleotide polymorphism or variation. Now this microRNA doesn't match. In this case, we will have more oncogenic messenger RNA and more oncogenes, and this one step toward the oncogenic transformation, right? Here is the opposite situation, much more interesting one. This is the tumor suppressor protein, and we need tumor suppressor proteins. They are very valuable, right? And there is almost perfect match of some of the microRNAs. We have actually thousands of microRNAs, right? But it doesn't fit. So this tumor suppressor protein is fine. It will be produced in sufficient amounts. In case of polymorphisms, one of the microRNA becomes functional, and they start working against this tumor suppressor protein. So we'll have less tumor suppressor protein. And this is a big step toward oncogenic transformation, much more important than more oncogenes. So less tumor suppressor is a big deal. It's really not good, right? So this is how polymorphisms may mechanistically, explaining mechanistically, one of the variants, and this is the common explanation, how they may influence development of various diseases, including cancers, right? Excuse me. Right. Now I want to tell you about the project and the students who work on this project. So I mentioned that, well, we started from the finding, and Holly Clifton, she was the first grad student. She started in 2018. We started with the finding we made before. I don't refer to this presentation. The American Association for Cancer Research. It was done just in 2018. We made the poster, and I started this work with the students at Dakota Wesleyan University. But what we found that these sites, microRNA binding sites, right? Well, let me show it here. These microRNA binding sites, they may have more than one polymorphism. For example, like this. This one polymorphism, there may be even more. The second one, and even the third one. And they're usually at low frequency. And we became interested in the combination of this polymorphism. And that's what the project Holly Clifton started. But it was very difficult, and she could not bring it to defense, so she defended on another aspect. And another aspect is what we have. We have multiple microRNAs that binds to one protein. In fact, each microRNA binds to hundreds of messenger RNAs. And each messenger RNA is regulated by the hundreds of various microRNAs. And what is interesting, some of the microRNAs, like miR34A, they may have more than one binding site on the messenger RNA. It was somehow known, but not much discovered. And I knew this. The Holly was working on the human MET oncogene, and we looked up. It was already time for her to defend, and we kind of huge work was done, and she was making alignments manually, right? We suddenly found that there are many more these multiple binding sites than we expected in human MET. And when I was searching the literature, I could see that no experiments were done on mammalian cells. Basically, no experiments. The experiments were done on some other cells. And the meaning of this multiple binding sites, and we call them homologous sites because they are. That means that they are a perfect match of what we have here, right? This multiple binding sites, this is the match of here, but the rest may be different for each site. So they are homologous, they are perfectly identical only for this part to guarantee, to ensure that microRNA may bind, but they differ in other areas. So then in this part, they don't have much. In this part, there may be some additional matches. Actually, the more matches, the better, right? So homologous, but anyway, they may work for the same microRNA several times. So why it's important? It's important because the homologous sites, we call them homologous sites, may enhance the effect of the same microRNA, and is present in the literature. So microRNA target the messenger RNA in two places, so the stronger effect, number one. Number two, if one of the sites is disrupted by the polymorphism, the second site may still be functional, right? And it may preserve the functionality of the microRNA, right? So microRNA still will be active. It's especially important for oncogenes, for oncogenic messenger RNA. If one site is disrupted, then the second site may still may allow microRNA to keep this messenger RNA under control, right? So that's what it was work done by Holly Clifton a little bit, and unexpectedly we found that, well, almost 1/5 of the microRNA that were targeting human MET oncogene, they utilized more than one site. And if you are talking about these repetitive microRNA binding sites, well, there were plenty of them. They were repetitive. And each of them, one. So this is the only one site that happens in once. These sites are duplicates, 81 triplicates, events 8 quadruplicates. So, and I should say that we have some data when single microRNA may have up to 50 multiple binding sites on the same messenger RNA. So that's interesting. We have such data because we are processing the data from the target scan database, and Summer, she knows about this because she was working, right? So I'm showing you just a little bit data of Holly Clifton, and we published the paper, unfortunately not in hierarchy journal because we need to publish it right before her defense. But anyway, so she analyzed there are non-repetitive conserved sites according to the database, that sites that are present in various biological species, and repetitive conserved sites and non-conserved sites. So they're all present, and these repetitive sites, they're pretty much, they're pretty much present. They are the factor. There are less repetitive sites than other sites, but they are present. And we decided that we need to study them, and it was interesting, right? And this is the publication. Yes, and the authors, the Holly Clifton, Verona, Georgia, and Summer Summer Stephenson. Now, I'm sorry, you are Summer Weeks, right? Yeah, well, I hope that we publish. And this it was a starting point. And now we are studying the sites. Before I go to Mirona, Georgia, we are working on this. We have the ongoing project. We even submitted grant, well, based on the site. And it will start almost from nothing, you know. I'm going to reflect every participant, and Dr. Zhong as well. I'm just presenting research. It's not about in depth of each, right? Now, the next project, so Mirona, Georgia, she's from Spain. Now she's back in Spain, right? And she's working, she's in PhD, right? What was her project? I told her that we were interested in multiple polymorphisms within the microRNA binding site. So this is a little bit non-canonical microRNA, but it has a very good stretch. It's not starting from the second or the first nucleotide. It's a little bit in the middle, but very good stretch, right? And how many polymorphisms we have here? Plenty of them. So this is the huge deletion that may inactivate the site totally, and this microRNA will never be able to find its site, right? There is another deletion, and here is also deletion, and there are, yes, and this red bricks substitution of nucleotides. So we start working on this. Finally, Mirona, Georgia was able to get to this, and I should say that these alignments were made before Dr. Zhong developed the first program, and I will explain you why. So it was done manually, and Holly Clifton was doing it manually. These beautiful alignments, we developed some approach how to get this map, how to cut out this map all on the computers, right, and to align and to calculate what's going here, right? So this is the essence of Mirona, Georgia's thesis for defense, and I can tell you, so there may be different variants. For example, so it's on the next page. The example, this polymorphism may be disrupting, enhancing, and neutral polymorphism. For example, starting with the first one, polymorphism number 1, right? In this case, as we can see, this first one means that A nucleotide will be substituted for T nucleotide. And here we have G. Normally, G doesn't bind to A. It's according to Watson-Crick, right? But G may bind to U, uracil. And this is, we are talking about the RNA sequence. Still, since it's genomic map, it's T instead of U, right? Biologists, you understand what I'm talking about, right? So according to, if it comes to genomic map, we never mention U, and they put it as standard. So, and what does it mean? G binds to U. Can it bind? Yes, it can bind. It's so-called wobble base pair, which is very typical for all RNA biology, and it has the same strength as canonical Watson-Crick binding, right? So in this case, we may have additional match here, and that means that binding of microRNA to its target will be stronger. And so on, so on. The next step, I'm not going to go through number 2, neutral switch from GU wobble base pairs to classical Watson-Crick GC. So GU was matched, polymorphism, nothing changed. Neutral switch, right? Now, where is disrupting? Number 5, disrupting. And 6, enhancing. Let's take a look here. Disrupting, number 5, what's happening here? Number 5 is, well, they're all put all 3 together. So this is the perfect match as it's supposed to be, but changes the presence of these polymorphisms will disrupt these links here. While polymorphism in this case may add additional link. So it's a huge project to study. And of course, what we wanted to calculate the probability of disrupting in case of multiple polymorphisms with known frequencies in the population. And here was help of Dr. Vitaly Voloshin, and he suggested to use the probability of the union formula, and this is the simple event when we have only 2 polymorphisms, coexisting polymorphisms, and they are not overlapping polymorphisms. So it was formula, but, and this is a different situation. We have overlapping, mutually exclusive events. This is the deletion, this is the polymorphism that shows here with a known frequency. This one frequency apparently not known. So it's a different formula. And Mirona had, 5 and 6 multiple polymorphisms. So this probability of union formula was really huge, and she was calculating herself with the computer. We would really need help. We need a lot of help from computing people, to put it into work, but it's deep. So it was a second project, and the results indicated that multiple polymorphisms may enhance binding, or they may even further disrupt the binding, increase the probability of the binding between microRNA and microRNA, or decrease the probability, right? And we were analyzing only those that enhance, those that disrupt. We were analyzing only those that disrupted, those that enhanced. It's a separate project, and a lot of computing need here. But it's interesting because it seems like what we know now is not just single polymorphism that may predispose us. It may be combination of polymorphisms in various messenger RNAs and microRNAs. Let's go next. Yes, this what, okay. Now this is about computing. This is the publication, I'm sorry, I'm going to France. This is the publication that was made with, by Dr. Zhong and his student, and the first student, Neil Purish Kumar. He unfortunately, tragically, I saw him only once in the office. He died just within days after he presented his work. So, and the program he created, it was called NEAL, Nucleotide Expression and Logic, a new computational approach to the analysis of microRNA-related nucleotide variations. So what we needed the very basic things. We needed to align, just like it was aligned here, right? Like it was aligned here. We needed to be able to align our microRNA to the microRNA. But all the alignment tools, classically, are based on similarity. That means you have T here, there should be T here, G, G. We needed align based on the complementarity. Those who took molecular biology with me and genomics, you probably understand what I mean, right? We need this alignment. We need alignment of microRNA. And such tools were available at the beginning of 2000. Then they were just removed because the smart people decided we need just to map sites. And already around 2014, in the forums online, they said, well, we need alignment. So Dr. Zhong took this project and made the first alignment tool, right? And it was working, and we were using this alignment tool. And Miruna did her thesis using this alignment tool, but we needed even more. So fortunately, Francisca, the student who worked with Dr. Zhong, graduate student, he took it as her Master of Science thesis, and she made this the image from the online software program. She made the tool that allows to align RNA sequence based upon the complementarity and to predict microRNA binding sites. And my genomic class surely knows what we are doing, right? We are working on this full steam ahead, and we are using her program here, right? This is the Summer Stephens, right, who is sitting here, her project, and this is, okay, and this is Caroline, and this is Travis. So there is something interesting happening here, and unfortunately, I cannot give this. It seems like these microRNA binding sites may be distributed unequally along the 3' untranslated region, unequally. We have this pretty good data with target scan, and I should appreciate the effort of Sama, who made a huge deal of work to collect this data. Yes, and I just want to highlight. We start analyzing. We got very nice data with the target scan. We start using Francis's program. We are using it now. Seems like not looks, but Travis offered the new macro just today during the class, so it's the last moment photos made a few minutes ago. And we are proceeding with project altogether. Hopefully, we will publish soon about this, right? What else we have? Now, Martin Buldo. He is a student from the physics. He is great programmer. He is working, his project is to combine the Francisco tool that is related to microRNAs to what we have with polymorphisms in the NCBI database, specifically in the NCBI database. And he is developing this tool and hopefully he will do something by his graduation, right? All right, so this is our chapter. We managed to get with our data to the chapter and all this after, and yes, this is the Robert Kaltenbach who worked on this. He did a lot of work and he made the first macro code allowing. He started work in 2020, and at first it was not macro code, just the algorithm how to work with the homologous microRNA binding sites. So you see this homologous microRNA binding sites. It was done by Holly Clifton at the very beginning, and we're still working on this, right? Yeah. Now acknowledgements. I'm coming here. So by the way, so this is. It looks like kissing cells. The cells are not kissing each other. They are highly malignant brain tumor cells. They're just dividing, yes, saying goodbye, but they look like they're kissing. So anyway, with love, acknowledgements to the Department of Biological and Environmental Science and Dr. Zeke Harden. I want to stress his contribution from the very beginning. And there were good times, there were not so good times, and this project was subject of survival at a certain point. And Dr. Harden, if not Dr. Harden, you would not have this research here. And Dr. Brian Helms, these first two students, Holly Clifton and Francisca, Holly Clifton and Mirona Georgia, they were students at the environmental graduate program, and they were working with me, right? Department of Computer Science, Dr. Zhong, we collaborate greatly, and I hope that we'll bring this collaboration to the new level, you know, educational level, one step more, right? I hope. And Dr. Vitaly Voloshin from the Department of Mathematics, he helped a lot with Mirona's project. And what we have here, Hudson Alpha Institute and Code project. I have to say, yes, that Hudson Alpha Institute runs his own bioinformatics project, Alabama-wide. It's so-called characterizing our DNA exceptions. So Michelle Morris, she was here, she was invited, and I know that somebody, after her invitation, right, to went there, and you will share your impression with Hudson Alpha Research Institute. So our project is different. They all work with proteins. We work with RNAs. Our project is acknowledged as a part of this code project, which is nice, and it was a little bit ambitious, you know, because they are very good. They helped a lot, you know, about bioinformatics, and they help a lot with making this course, a lecture course. So thanks to them. Dr. Mikhail Kashlev, Senior Investigator, RNA Biology Laboratory, National Cancer Institute, NIH, and Dr. Astrid Hase. They are supportive. They issued the letter of support when we tried to submit this first grant, which was arrogant. They are both top world-known investigators. How they become supporters is because of students. Dr. Mikhail Kashlev got one of the students from Dakota Wesleyan University, very good students. They were so much impressed that they are collaborating. Dr. Astrid Hase, it's Francisca. She works with her, and they're so happy with our students that they offer the letters of support. So basically, this is all, and I want to thank everyone for coming here and to getting an idea. And if you have any questions, yeah, I'm ready to answer. Pretty complex, right? Not too many questions, maybe. So we are doing ongoing research with the current class, genomic class, and guide independent research, and we are pushing this project for the analysis. What we have, we view two small publications we are publishing in the journal that's called Biomedical Journal of Scientific and Technical Research when it comes to any programming tool, whether it's macro code or even the software. And it's very, very inconvenient to publish because, well, it's low ranking, but it's peer-reviewed and it has some impact factor. And we hope to submit the publication after we published our chapter. We immediately, it was online, and we immediately received probably automated invitation from the journal Cells based in Basel to contribute to the special issue of the special issue. It's about polymorphisms they're preparing, and this is a good journal with the impact factor, and they making usually special issues rather than presenting them. So hopefully we'll submit by December 15, and every participant will be in there. Any questions anyway? Difficult project, very difficult, very unusual, I would say. and it was doable here when I was leaving research as a postdoc and starting my teaching. And first at community college, I knew that I would better to use some kind of theoretical biology project because it would be available in any teaching institution. Well, it varies. I could never expect that it would go well to such an extent. And of course, thanks to computer science, without you folks, nothing would go. We cannot do research by doing it manually because it's simply, it's not fashionable. They said, well, you did manually. Results, maybe results, but tools are needed. Okay, thank you so much. Thanks, Office of Sponsor Program. Thanks for this great opportunity to present this, and thank you for coming here.
Trojans Talk Research is a monthly spotlight on the research interests of choice faculty presented by Troy University's Office of Sponsored Programs.